Hey, my name is Erica Fellin, and today I'm going to be talking to you about the effects of ticks on community assemblages of ectoparasites in deer mice. So, a community is a group or association of populations of two or more different species, occupying the same geographical area at the same time. For my research, I focused on external or ectoparasites, such as fleas, ticks, and mites, that occupy the same host as a community. In this case, the community was the deer mouse. This small mammal has a wide geographic range across North America and is found in many different habitat types. It is a viable host to a number of ectoparasite species and has been found to be susceptible to several zoonotic pathogens. These mouse hosts also act as islands, allowing for certain ectoparasites to transfer from one host to the next, allowing for transmission of these pathogens within a population. Ticks, particularly hard ticks or ixodid ticks, are arguably the most medically important ectoparasites in North America that feed on these mice and transmit pathogens. Ixodid ticks have 3 life stages, where they feed on the blood of their host to reach their next life stage. The larval and nymph stages are when they tend to feed from smaller mammals like the deer mouse, and it is during these life stages that they tend to acquire pathogens from their hosts, which are considered reservoir hosts. Once ticks have acquired pathogens during immature life stages while feeding, they will move on to larger mammals, including humans, and feed from them, potentially transmitting the same pathogens they've acquired from their previous hosts. Knowing ticks acquire pathogens at their immature stages from reservoir hosts like deer mice, it is important to consider their relationship with hosts at these stages. It should also be kept in mind that there tends to be several parasite species infesting one host at a time, many of which can transmit pathogens and physiologically affect their host. In order to better understand the spread of these pathogens as ectoparasites' geographic ranges expand with climate warming, we need to recognize how parasites interact with their hosts and their parasitic communities, as these interactions can influence how a pathogen is transmitted. Several different species of ectoparasites, including ticks, fleas, and mites, are found together among their host. Here, the host is considered an ecosystem or habitat itself to these parasites within a community. In this case, 2 scales must be considered. The macrohabitat, which is the ecosystem the host is inhabiting and where it interacts with other organisms, as well as the host itself, acting as a microhabitat, where the ectoparasites interact with each other. One study has found that parasite community assembly and structure is affected by the individual host as well as the host population, where environmental factors at both the micro and macrohabitat scale affected community assemblage. Across the macro and microhabitat scales, habitat partitioning has been found to affect community assemblages. And this is important to consider as ectoparasites tend to feed from their host in different niches or attachment sites of the body. Ectoparasites tend to prefer exposed areas on the skin on their host to feed from, including the ears, open wounds, and genital areas, where they will opportunistically feed from these exposed spots. For my study, I focused on the relationship between deer mice hosts and 2 tick species that parasitize them, blacklegged ticks and wood ticks. Wood ticks and blacklegged ticks are both found in southern Ontario and can be found in similar habitats. Wood ticks have been established in Ontario longer than blacklegged ticks and have expanded their current geographic range further. However, as the global climate warms and more habitat becomes suitable for both species, both of their ranges are projected to expand further north. The map here shows the predicted suitable habitat for wood ticks by 2050, in which wood ticks are likely to have established populations well into northern Ontario. Blacklegged ticks are also expected to establish in northern Ontario in a similar time frame, as seen in the map to the right. Since both tick species can transmit zoonotic pathogens to several host species, including humans, they are important parasites to monitor. Wood ticks can transmit Rickettsia rickettsii, the causal agent of Rocky Mountain spotted fever, while blacklegged ticks can transmit Borrelia burgdorferi, the causal agent of Lyme disease. To get a better idea of how deer mice may be affected by different tick prevalence, I chose to sample from 3 different study sites across a latitudinal gradient that represent varying levels of establishment for both of my focal tick species. The geographic range of deer mice coincides with all 3 of these sites, though the parasite communities of these populations vary. The most southern site, Long Point, has both species present, while the middle site, Queen Elizabeth Wildlands, has only wood ticks present. In the most northern site, Algonquin Park, neither species has been known to colonize this area. By looking at these present distributions of tick establishment, it was my goal to get an idea of how things may change in the future as these tick species ranges expand. My objectives for this project were to compare deer mice from habitats with varying tick prevalence to determine differences in species distributions between sampling population sources, ectoparasite attachment sites on hosts, and co-occurrences of ectoparasites on hosts. I hypothesized that if ticks were the dominant ectoparasites in a microhabitat, then fewer other parasite species would be found, since ticks are occupying preferred attachment sites. Here I expected that tick presence would alter parasite community assemblages. Although the provincial parks I trapped in were of varying size, I tried to have a consistent trap effort across sites by having 3 trap lines per site, where traps were set in mixed forest areas to have as similar habitat as possible across sites. In order to account for changes in seasonal patterns within my field season, as certain parasites are more active at different points and floral and faunal patterns change over the course of the summer, I traveled between the 3 sites, collecting data 1 week at a time from each location. I traveled from the most southern site to the most northern site, and then back again, traveling from one site to the other each week between May and August of 2019. When collecting mice from traps at each site, I identified mouse species morphologically, then aged, sexed, and weighed mice on site. I collected all ectoparasites that could be found on them using forceps for 60 seconds per host. In lab, I identified my tick and flea species, which were later confirmed by Dr. Robin Lindsay and Dr. Terry Galloway, respectively. 2 distinct mite groups were also identified by Dr. Heather Proctor. Although these mites could not be ID'd to species, Dr. Proctor was able to determine that there were 2 taxonomically distinct mite groups, and I will refer to these mite groups as trumbiculid mites and neotrumbiculid mites. While collecting ectoparasites, I also surveyed where on the host body they were found. I divided where I found ectoparasites into 5 separate attachment sites: the head, the ears, the middle of the body, the posterior or back of the body, and extremities. And for extremities, I included forelimbs, hind limbs, genital area, and the tail altogether. The species distributions across population sources found in this study varied, though the same flea species was found at all sites. In Long Point, or LP, all ectoparasites but the trumbiculid mites were present. In Queen Elizabeth Wildlands, or QEW, blacklegged ticks and neotrumbiculid mites were absent, and in Algonquin Park, or AP, both blacklegged ticks and wood ticks were absent. Statistical methods were conducted in R, where I performed several ordination analyses. This included an unconstrained correspondence analysis to determine what niche or attachment site on the host ectoparasites tend to occur based on presence-absence data. A canonical correspondence analysis, constrained by environmental factors, was conducted to determine what variables may be affecting ectoparasite species distributions. Environmental factors included microhabitat variables, such as the host body mass, sex, age, and reproductive status, as well as macrohabitat variables, which were population source and session in which the sample was collected. A non-metric multidimensional scaling, or NMDS model, for ectoparasite species distributions across population sources was also analyzed. This model used a dissimilarity matrix to determine patterns across population sources. Finally, I performed a co-occurrence analysis based on presence-absence data across individuals and then also across population sources. When observed co-occurrences were extremely low or extremely high relative to the expected co-occurrence, these associations were considered to be negatively or positively co-occurring, respectively. For the unconstrained correspondence analysis, comparing microhabitat niches of ectoparasites, it was found that both mite groups and blacklegged ticks were found exclusively on the ears. On axis CA1, all ectoparasites, except the flea species, were found to have positive scores, where fleas were the only ectoparasite that were found on the back of the mouse or extremities. Meanwhile, wood ticks were commonly found on the ears, but not exclusively, and were infrequently found feeding from the middle or the head of the mouse. The constrained correspondence analysis assessed environmental factors that may be affecting ectoparasite community distributions across hosts. The only environmental factor measured that was significantly affecting this distribution was population source. With that in mind, the NMDS model compared ectoparasite distributions across population sources, where it was found that there was the greatest overlap between Queen Elizabeth Wildlands and Algonquin Park. Long Point and Algonquin Park had the greatest dissimilarity in species distributions, where there were no overlapping ectoparasites found on hosts. So even though neotrumbiculid mites were found on mice in Long Point and Algonquin, They were not found frequently on long-point mice, and were not found on hosts that also had black-legged ticks or wood ticks present. Looking across all hosts independent of the population source they came from, several different co-occurrence patterns were found. Black-legged ticks and wood ticks were the only pair to have a positive co-occurrence. And both tick species had negative co-occurrences with trumbiculid mites and fleas. Wood ticks and neotrumbiculid mites had a negative co-occurrence, as they were never found on the same host, but were expected to, whereas black-legged ticks had a random co-occurrence with these mites. All other associations between fleas and the mite groups were random. However, when the analysis was constrained by population source, all co-occurrences were found to be random. It's difficult to determine if this result is meaningful though, as the algorithm that calculates expected observations between species pairs also considers the number of sites in the analysis. And for each population source separately, the sample sizes were much lower, and so the power for these analyses was diminished, since the sample sizes were not as robust. Going back to habitat niches across microhabitats, my results suggest that the most distinctly different species distribution was for fleas. Flea ecology is different from the ecology of mite and tick species, which are both from the subclass Acari. While mites and ticks attach to a host and feed from one spot, fleas tend to move freely between a host and its nest, and do not necessarily rely on one host per life stage. This may be why they were found outside the typical exposed areas where you would expect an ectoparasite to feed. Both mite groups and black-legged ticks were found exclusively on the ears of their hosts, but were not found together on the same host, though there were instances of trumbiculid mites and wood ticks occasionally found in the same niche. Across macrohabitats, the greatest species distribution overlap was between Queen Elizabeth Wildlands and Algonquin Park, where black-legged ticks were not present. At the same time, there appeared to be no species overlap on hosts between Long Point and Algonquin Park. And for ectoparasite co-occurrences, when all hosts were considered independent habitats, distinct co-occurrences were found, suggesting that certain pairs of parasites are likely to co-occur together or exclude each other. Specifically, mites and ticks tended to not occur together on a host despite having the same preference for attachment sites, which could suggest that there is competition occurring between these species for hosts, though further testing is required to truly demonstrate this. Especially since all occurrences were demonstrated to be random when microhabitats were constrained by their population source, though this may have been due to a power issue with the algorithm. Overall, field site was shown to be the only environmental factor affecting species distribution, which makes sense not only because of the difference in habitat, but the difference in tick prevalence at each site. On individual hosts, fewer other parasites were found when ticks were present, especially mite groups that were found in the same niches or attachment sites as ticks on hosts. And this could potentially be happening because tick occurrence alters community structures, as both tick species were found to be negatively co-occurring or randomly co-occurring with fleas and mites, but positively co-occurring with each other. Although, depending on constraints to analyses, this observation could be unfounded. In conclusion, I found that there was a difference in community assemblages when ticks were present in an ecosystem versus absent from one, though the macrohabitat that the host comes from plays a major role. And there was a correlation between population source and tick prevalence, though I didn't measure any environmental factors from these population sources, so it is unclear what exactly is influencing this correlation in community structures. However, these results should be taken into consideration when monitoring the geographic expansion of the ticks ranges, as deer mice and their parasite communities have been shown to be affected differently depending on the prevalence of ticks in their ecosystem. Further consideration must be given on how the ever-increasing prevalence of these tick species will affect ecosystems that will soon come into contact with them. Queen Elizabeth Wildlands is expected to have black-legged ticks invade in the upcoming years, and soon after, Algonquin Park will be invaded by both tick species. As naive hosts encounter these tick species, it is important to understand how tick prevalence will affect ectoparasite communities and the microhabitats they share. But at the same time, it is equally important to consider how the macrohabitat will be affected by these interactions, as these ticks expand their range, thereby expanding their pathogen transference capability. Thank you for listening to my presentation, and an extra special thank you to all the people that made this research possible. Let me know if you have any questions.